Good evening, everyone. My project focuses on finding effective treatment options for a deadly type of skin cancer called melanoma. With over 100,000 new cases this year, melanoma is the most common cancer in young adults. It is caused by the uncontrolled growth of melanin-producing cells of our body called melanocytes. Melanocytes, like many other cells, express receptors on their cell surface, which works very much like a TV antenna. It receives signals from the outside environment and transmits them to the inside of the cell, which allows the cell to grow, move, or even undergo cell death. This happens when a cell receptor gets bound by signaling molecules. Cysteine leukotrienes, or CysLTs, are signaling molecules released mainly by blood cells and are known to enhance inflammation, leading to chronic asthma in humans. They act via two receptors, CysLT1 receptor and CysLT2 receptor. Since inflammation is known as a major driver of cancer, we wanted to investigate the role of these two receptors in melanoma progression. We first found out that both these receptors, when activated by CysLTs, are causing increase of melanoma cell growth and metastasis. Therefore, we speculated that if we block these receptors, we could target melanoma progression. Now for the good part. Blockers of both these receptors are already very well established in the field of asthma. Singulair, which is the blocker of CysLT1 receptor, is already FDA approved for asthma treatment, whereas base CysLT2R, which is the blocker of CysLT2 receptor, is currently under FDA approval. So here we are trying to repurpose these two drugs for the treatment of melanoma, and thereby we are eliminating all the side effects of a typical chemo drug. We first treated our melanoma cells with both of these blockers and saw a very significant decrease in the melanoma cell growth and also their migration capabilities. We then tested this on mice by injecting these melanoma cells to mice that were genetically modified to not express either of these receptors throughout their body, thus resembling an inactivated receptor. And the tumors formed in these mice were much smaller in size compared to the tumors formed in a normal mice that expressed both the receptors. These tumors also did not show any signs of metastasis. Therefore, our future experiments are focused on finding the exact dose and efficacy of these blockers for melanoma treatment. Thank you.